Bienvenidos a La Oveja Eléctrica, un espacio para explorar lo que la ciencia nos dice sobre las fronteras del conocimiento, de nuestro propio cerebro.
El olor del conocimiento siempre despierta curiosidad.
Eso es lo que hizo que Linda Bock, la Premio Nobel de Fisiología en 2004, se interesara en descifrar el enigma del olfato. Bock, thank you very much for this interview for Channel 22.
Thank you for having me.
Since childhood, you liked puzzles. Tell us about the puzzles you tackled on your research and how the brain recognizes and remembers 10,000 different others. Because this is a puzzle, how this can happen.
First, how can we detect so many different molecules, 10,000 or more different molecules with our noses? So it seemed like there must be proteins in the nose that could detect these chemicals that would be floating in the air. They're volatile chemicals. They're all small. They enter the nose when we breathe or we sniff a flower. They must be recognized by something in the nose, a protein on a neuron, and then that neuron must send signals to the brain. And it seemed that there must be more than one of those proteins in the nose because you had to recognize different molecules, different chemicals, and distinguish them. But we had no idea how many there might be, no idea how to find them. People were looking for them, had been looking for them, but they hadn't found them. So what I decided to do was to look for genes encoding the proteins. You know, the way it works is that you have one gene that encodes one protein. Like you have an insulin gene that codes for the insulin protein. How is the brain organizing this information so that it can make some sense of it? There are neurons in the nose, millions of neurons in the nose, and each one uses only one receptor. So are there spots in the nose? Are all the neurons with the orange receptor at one spot? And then there's another one for the lemon receptor? Well, it turned out that that wasn't the case. They're really scattered.
It was a surprise for you to find it?
Yes, we found that first we found that each neuron used only one receptor. And then we found that neurons with the same receptor, like one of the receptors for orange, were scattered in the nose. There was no pattern. It seemed random. And so then we looked at the next level in the olfactory bulb, which is above the nose. And the neurons in the nose send these long extensions called axons to the olfactory bulb. And we were very surprised to find that the axons of all of those orange receptor neurons went to the same place, and they formed a spot in the olfactory bulb. So for every receptor, there was a spot. And the spots, this was looking at mice, we don't know about humans, but we suspect that it's probably the same.
The same mechanism.
The same. So, and we found that the spots had the same place in different animals, different mice. So the orange receptor spot was here, and below that was the one for almond smell receptor, etc. So that was quite astounding to us. We still don't understand. There are many mysteries that remain to be solved, but one is how does the neuron choose one receptor out of 1,000 receptors and only use that one? That's not known. And how does the neuron know to connect to the right spot in the brain?
In literature, you can see, for example, how some essayists would say the evil has an unmistakable odor. And not now, but the direction is to really tackle these mysteries of how we smell if something is dangerous, how we smell if something is very attractive.
There are many challenges for young people in science, And I hope more young people will choose to go into neuroscience to solve these mysteries.